So I wanted to welcome you all to our 10-year anniversary celebration. It's great to see many new faces also in the room. My name is Tarek Fadel. I'm the Associate Director of the Marble Center. And before introducing Sangeeta, I want to say a couple things about the Marble Center. So when I joined MIT about 10 years ago to work with Sangeeta, I was and continue to be really inspired by her vision to build this center as a hub for nanomedicine at MIT. The Marble Center is more than just, excellence in research and whatnot. It's also intentional in its programming and its approach to building a community mainly as well. You'll hear that from Sangeeta shortly. And of course, this couldn't have been done without the incredible support from Kurt Marble, who's with us this evening. Kurt, thank you for believing in our vision, and thank you for your generosity and your support. So now I wanted to introduce Sangeeta. Sangeeta is a John J. and Dorothy Wilson Professor of Engineering, Director of the Marble Center, a member of both the Broad Institute and the Wyss Institute, and an investigator at HMI. She also leads the MIT Royalty Pharma Faculty Founder Initiative. Many of us know Sangeeta as an inventor, professor, and entrepreneur. To me, she's first and foremost an incredible mentor who really pushes all of us to be creative, caring, and forever learning. Trained as both a physician and engineer, her research leverages miniaturization tools from the computer industry for medical innovation with applications in liver disease, cancer, and infectious diseases. Sangeeta and close to 100 trainees have contributed to more than 65 issued or pending patents, launched multiple biotechnology companies to improve human health, and published more than 230 peer-reviewed papers. She has won numerous awards, but just to list a few, she was the 25th person in history to be an elected member of all 3 U.S. national academies, that is the National Academies of Science, National Academy of Medicine, and the National Academy of Engineering. She has been honored with the Lemelson MIT Prize, the Heinz Medal, and the Othmer Gold Medal. She also currently serves as a trustee at Brown University and director at Vertex Pharmaceuticals. Please join me in welcoming Sangeeta.
Thank you, Tarek. You promised to keep it short. Hi, everybody. Welcome to our anniversary, our birthday, our celebration of the Marble Center. We're so excited to really be here with you and have a chance to reflect on our journey and our goals for the future. Before we start, and in the rare chance that I forget, I want to thank Tarek, our Associate Director, for really being the brainchild and the thought partner behind all of this. So thank you for bringing us together, and thank you for everything you've done so far. Okay. So I think many of you think about nanotechnology, but I wanted to sort of emphasize that it has really 2 origin stories, and I don't think we always think explicitly about that. On the left here, you see the semiconductor microfabrication origin story. These are the things inside all of your iPhones, and really the drive for miniaturization has come from the wish to speed computation, really not for nanomedicine, right? But we're borrowing that advance for nanomedicine. And on the right, what you see is the origin story of solution-based aggregation of molecules. These are quantum dots, for which there was a Nobel Prize at MIT in 2023. And these really cool materials have nanoscale physics, electronics, magnetics, optics. These ones are all the same material. They're being illuminated by the same wavelength of light, and they glow different colors depending on their size. So they're really interesting physical properties that happen when you shrink these materials down to the nanoscale. And what we've been interested in as a community is really marrying this nanoscale, the opportunity to make materials this small, with the opportunity to talk to biology. So here I have a molecular ruler, and you'll see all the way down from 1 nanometer down, we're in Boston, to the size of a baseball. And a single cell is about 10 microns, a virus is 100 nanometers. We have materials now that can communicate with these structures. A single receptor is 10 nanometers. And so now we have lipids, polymers, plasmonics, magnetics, materials that can go into the body and communicate listen and respond to these materials. And so what's been really exciting as a community over the last 10 years is we've discovered some new things about nanoscale biology. The community was really interested in nanoscale physics, and the properties of these materials have been emerging and being exploited, but we learned that if you put them in the body, they do things depending on their size. So for example, we've learned that they traffic different places depending on how big they are. 5 nanometers will come out in the urine, larger things will go to the liver, decorated things can go to different organs. We've learned that the shape of these materials can dictate whether they're taken up by a cell or not. We've learned that we can make these materials actually respond to the biological environment and change their properties. And we've been able to harness some of this to deliver genes, to excite the immune system in our favor. And I'll show you some specific examples of the ideas that we've been really excited about. So I just want to emphasize that nanoscale physics has now also given us nanoscale biology. And together, there's so many opportunities that we can now exploit. Just as one example to sort of think through how fast things are going, fields are converging. So on the top here, we have the field of miniaturization that I alluded to, the mainframe computer, the first IBM computer, all the way to the present day where the current hardware chips have 3 and 4 nanometer nodes, right? Incredibly tiny structures that we can build with nanofabrication. And at the same time, elsewhere in biological research, we were collectively working on RNA. And you can see when it was discovered, right about the same time, when the first RNA therapies got approved, when they got packaged in LNPs, when they got it put into millions of people for COVID. And then most recently, for those of you who follow the news, about a year ago, there was an N of 1 therapy for baby KJ that was done with base editing. So these two timescales have been kind of happening alongside each other. And they're coming together now in this moment. And I could draw the same thing for genomics, for stem cell biology, for proteomics, for spatial technologies. All of the things that are happening in biomedicine are overlaid on this accelerating moment. And now we have AI, right? The hardware enables software. So there's just so much coming together now that it's an incredibly exciting time. And for the trainees in the audience, like, you're here at just the right moment. So together we are a community of faculty that have come together with our labs to really ideate together and be ambitious about the future together in a joyful, safe, creative way. And that's the community that we really have created, and I'll show you some of our metrics. As I said, Tarek runs the center, and you'll meet most of our faculty today. Our goals when we started 10 years ago with Kurt and Kathy's support were to build a community, to train the next generation of leaders, and to translate inventions that maybe were considered high risk or futuristic towards patients and into the future. And here's just some, by the numbers assessment of our progress. So scientifically, we've published a lot of papers with a lot of citations. We have given many, many research seminars. These are internal seminars where we share unpublished data, and many new collaborations emerge from those. We've given annual symposia. We have a presence at the Gordon Conference in nanotechnology. And then we've really tried to breed the next generation of talent. So we have 491 alumni, postdocs and grad students. They themselves have become faculty now across the country and across the world, both in universities and in academic medical centers, treating patients. So it's been really exciting. Our goal in bringing them up as part of our community is to make sure that they're ambitious and interdisciplinary, and also that they're leaders. This is a field that's evolving. It's not without geopolitics, and they need to be able to be kind of ambassadors for our field. And finally, we think we're beginning to have translational impact. Biomedicine takes a long time to get to patients, but we're really getting there. We have inventions, 23 startups, 8 IND-enabling studies. Those are the regulatory packages that you do before the first-in-human studies. And we've raised, these startups have raised additional funding based on our sort of high-risk accelerator grants. So we believe that we're starting to have an impact and will grow even more over time. This is a visual map of where all our alumni are all across the world. So you can see we're really touching all corners. And I just want to give you a couple of examples before I wrap up. And so just to set the context for these examples, this is really a time course for cancer that we're collectively trying to conquer at the Koch Institute. And the idea is that anywhere along this journey is an opportunity to intervene. Whether you're assessing risk for cancer, whether you're detecting early when it's more curable, whether you're diagnosing it specifically so you can treat it selectively, whether you're monitoring treatment response or resistance, and then eventually whether there's a recurrence and you're going to change treatment. So these are all areas, opportunities for us to intervene with nanomedicine inventions. And we have projects that we have funded on both sides. On the detection side, I'm going to show you a couple of examples. Here's 3. And then also on the treatment side, the idea that we can package materials, we can deliver genetic medicines, or we can excite the immune system. So really anywhere along this landscape is something that our trainees should be thinking about. So just to give you a couple of concrete examples, one nanoparticle diagnostic enhancer that was invented in our lab together with Victoria Edelsteinson, who you'll hear today, and Chris Love, is the idea that you could do what they call a liquid biopsy. So you take a blood sample from a patient or a prospective patient and you look for shed molecules from the tumor in the DNA. And our observation together was that the liver was actually clearing a lot of these. So when you take a blood draw, there are not very many left for you to sequence. So we took advantage of the known nanoparticle observation, which is that if you give a decoy nanoparticle, you can distract the liver for a few hours. People had done that for therapeutic reasons, and so we did it here for diagnostic reasons. So we came up with a decoy nanoparticle that prevented them from eating up that DNA for a few hours. And now when you take a blood draw an hour or 2 later, you have 100 times more signal, more mutations, more sensitivity. This is a paper we published together last year. It became a startup. Our students are in the startup, and so we're kind of on our way, we hope, to the clinic in about a year. This is an example from Bob and Dan of really using AI, machine learning, and neural networks for optimizing delivery. Those lipid nanoparticles that package the nucleic acids that I talked about, whether they're mRNA or siRNA, or even base editors for editing the genome, need to be optimized so that they don't go to the liver and they go exactly where you want them to go. And this is a project where they built a neural network with variable lipid components in it, and then in the computer, in silico, they could look through 1.6 million lipids instead of doing all the experiments and identify the optimal lipids to deliver genes into the lung. And here you can see, for those of you who aren't experts at looking at this, like green is good. Green is more delivery into the lungs. Then another example that sort of sits at what we call theranostics. This is out of Angie's lab. This is an amazing project that I think came from her and her group's creativity, where she is really great at imaging things. And when she gets told something is invisible, she says, everything is visible at some wavelength. And this is an example of she and Jeremy visualizing lymph nodes at about, I think it's short short microwave, so about 1,000 nanometers, so still in the nanoscale. And when you shine that light on these tissues, lymph nodes that were invisible become visible. And Angie and Jeremy, her student, learned together that actually before doing this visualization, this instrument that they now have in the hospital, surgeons would actually feel for lymph nodes. And now you can do this quite easily and quite quickly. Jeremy started this company, went to the White House to share it. It's now from idea to clinic in just 3 years, which is incredibly fast. And it's already in 15 US hospitals. So that's been really exciting to see a brand new idea come into the clinic. And finally, an example of therapy from Paula Hammond. This is the idea that we need to activate the immune system. Activate the immune system to be on our side and fight cancer. And in some cancers, this has been quite successful clinically. You will have heard of this, for example, for melanoma. But in other cancers, like ovarian cancer, it hasn't been successful. And we call that sort of colloquially in the field a cold tumor. So even if you activate the immune system, somehow it doesn't attack the cancer. And what Paula and her group have come up with is a way to make nanomaterials that will bind to ovarian cancer cells. She did that through a screening process here at the Broad, and then deliver a stimulator, IL-12, that will stimulate the immune system locally. And you can't give it systemically because it's not safe to give all over in the bloodstream. And those two manipulations, binding to the ovarian cancer cells and delivering IL-12, turn the cold tumors hot. And in this study, which she just published, she had 100% survival in mice after 6 months, which is an incredibly exciting result. Okay, so that's just a smattering of the kinds of things that we work on. This is a visual of how we're progressing to the clinic. These early stage projects come in the back end and these preclinical studies, and then we progress them, we spin them out, and we hopefully get them into the market. And so what I think you can look forward to from us in the future is more coming out and more coming in. So just more throughput through this incredible pathway. And finally, I just want to mention that we are trying to lead and bring the community into new areas all the time. We issue special requests for proposals for areas that we feel are understudied. A few years ago, we did one in global oncology. And the idea here was to have access to these nanomedicines all over the world. We are interested in Healthy Planet and the role of nanomedicine in climate change. And we're launching this summer this specific one called Insight. And the idea here is, as I said earlier, to make the invisible visible and to pull in community members all over campus that may not even be in our center who are really interested in imaging at the nanoscale. So if you're not in our center and you want to connect to us, please look out for that in June. Finally, I want to say thank you. Thank you to all of you for being part of the community and for giving your ideas and your time and your enthusiasm and your energy. To Tarek, who I thanked already, and to Kurt and Kathy, who have been on the journey from the very beginning. We really are following in the footsteps of Bob Langer and Ralph Weissleiter, who had coalesced the community in the Cancer Center for Nanotechnology Excellence before we existed. And really laid the foundation. And one of the reasons we needed to be here is that federal support went away. And we really felt it was important. There was so much left to do, as I showed you, with those converging arrows, that we really built on that foundation. Tyler, who was the institute director, who helped us get started, and everyone else in our community, the trainees, the faculty who are here today, the alumni, clinical and industrial collaborators, and our incredible scientific advisory board. So thank you for being here. And with that, I'm going to introduce our next speaker, Bob. I should start by saying Bob is legendary. And in some ways needs no introduction, but nevertheless, it's fun to do. Robert Langer is one of 9 institute professors at MIT. Being an institute professor, for those of you who aren't at MIT, is our highest honor. He has published prolifically, and his articles have been cited over 475,000 times. His H-index, which is the way we measure sort of academic impact in some circles, is the highest of any engineer in history. His patents have been licensed or sublicensed to over 400 companies. He's a co-founder of many companies, including Moderna. He holds 45 honorary doctorates, so he's doctor to the 45. Received over 220 awards, including both the U.S. National Medal of Science and Technology and Innovation, and he's one of only 3 living individuals to have done that. And he's been elected to all the national academies, medicine, engineering, science, and inventors. So we're so grateful to have him part of our community and here today. Thanks, Bob.
Thank you. Thanks so much. Thank you. thank you, Sangeeta. Thank you, Kurt. And really, it's an honor to be able to present a talk. Sangeeta said, could I present something on nanomedicine, past, present, and future? And of course, she actually did an incredible job, much better than I'm going to do. And I thought I would just sort of share with you my personal experience. But really, in a way, what I'm going to tell you is what happens when somebody takes an unusual path, comes up with ideas that at the time make no sense to most of the academic or business community, tries to get funding and can't, but you keep trying, and then as we can see, because of so many people that have been involved, that now it's led to products that are really changing the world and will continue to do so. So for me, I got my degree in 1974 in chemical engineering, and all my friends went to oil companies, and I just didn't want to do that. I was looking for some way that I could do something that I felt would help people. And I ended up getting a job at a hospital, a children's hospital, working with a man named Judah Folkman, who was a surgeon, but also a cancer biologist. And I was the only engineer there. And his idea was that if you could stop blood vessels, maybe that would be a whole new way of stopping cancer. And he was widely criticized. I knew nothing about biology. I thought it was a great idea. And I thought it could make a big difference if I could even make a partial contribution. One of the biggest challenges, and maybe the biggest challenge in trying to solve this problem, was there wasn't any good way to study blood vessels, because there's always background blood vessels. But there's a couple of places where there's none or very few. One is a cornea, another's a choroidal membrane. So our idea was that maybe we could put a tumor, this is back in 1974, in those sites that would cause blood vessels to grow. And then you wanted a tiny little vehicle, like a nanoparticle or microparticle, that could deliver large molecules and for a period of time. And that had never been done before. And in fact, in 1974, there was only one company in the world, Alza, trying to even deliver molecules of any size. So Dr. Folkman went out there, he was on their board, and asked them about that. And they all said, well, that really can't be done. They had Nobel Prize winners in chemistry on their board, too. They said it just can't be done. The molecules can't get out. They're too big. Or if they got out, they'd get out in a second, because it's very porous. So, but if we were going to solve the blood vessel problem, we had to solve this problem. So I spent several years in the laboratory experimenting with different materials, different techniques. I actually found over 200 ways to get this to not work. But eventually, I was able to make little micro or nanoparticles. You see a picture of one and one cut in half. And we published in the journal Nature, actually 50 years ago, that you could use this approach to deliver molecules of any size, even do it for nucleic acids like DNA or RNA. But when I first presented this work, which was actually even before this paper came out, it was really ridiculed. I was 27 years old, and I remember talking to this audience of very famous chemists and chemical engineers. And when I got done explaining this, and I was super nervous, they all told me that they didn't believe anything that I had said. And in particular, they said the molecules, again, were too big, and the solvents we were using, like methylene chloride or ethyl alcohol, would destroy whatever we put in. And the consequence of that is when I tried to get grants, my first 9 were rejected. It was almost impossible. When I tried to get faculty positions, no chemical engineering department, which was my main department in the world, would hire me. I got rejected from every single one of them. But Dr. Folkman introduced me to the head of the nutrition department, Nevin Scrimshaw, who was a benevolent dictator kind of department head, meaning that he liked me, so he offered me a job, but he didn't bother to ask anybody else in the department what they thought. And that would have been okay, except for one thing, which was that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. It's unfortunately true. I still remember back in 1979, And this is a good friend of mine, Mike Marletta, who was a colleague of mine then. Mike later became head of Berkeley's chemistry department and head of Scripps. But then he was at MIT as a young professor, and he was just reflecting on a dinner that he and I went to. And I'll just read you what he said. He gave this at a speech a few years ago when I got some award. He said, well, one evening I went to a faculty dinner at a Chinese restaurant. It was actually just a few blocks away, with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And Mike said he thought he was in a Fellini movie. At any rate, I kept working on it. We actually were able to use this to isolate the very first blood vessel inhibitors. And one of the things, and now there are many, Avastin, one of them, is one of the best-selling biotech drugs in history. Genentech did that work, building on some of the things that we did. But going back to the 70s, Dr. Folkman and I talked about getting a patent, which, again, might seem quite common today. But then, 1970s, Children's Hospital never had a patent. So we filed for a patent for the first time, and it got rejected 5 years in a row. And I remember one day the head of the tech transfer office coming to see me, and he said, well, Bob, you are wasting so much money. He said, just quit. But I don't like to quit. So I started to think, how could we convince the patent examiner to allow the patent after these 5 rejections? Legally, of course. And science really wasn't working. So as I told you in the beginning, when I first started doing this, everybody told me this was impossible. It could never work. So I went to do a literature search to see who wrote about our work, and I found a number of articles. I'll just show you one. Here is, he said, 5 of the really top material scientists in the 1970s describing this field. And they said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released because of their extremely small permeation rates. However, Folkman and myself have showed some surprising that were results that clearly demonstrate the opposite. So I like that word, surprising. I showed it to the people at the hospital. They showed it to the examiner. He said, gee, he had no idea. But he said, to tell you what, I will allow this patent if Bob Langer can get written affidavits from each of these 5 people that they really wrote it. So I wrote them, and they're nice enough to write me back they really wrote it. And we ended up getting this very broad patent after those rejections. And then, like I said, this was 10 years after I started, and I really wanted to see the work I did make a difference. And so, anyhow, finally, after all this time, I'd get calls from several multi-billion-dollar companies and said, well, gee, they'd like me to consult for them, and they'd like to license the patents, and they'd work on it themselves. And they gave me a big grant, and I was very excited about that. But after a year or two, they just both gave up. And so Alex Klebanoff, another one of my colleagues at MIT, said to me, Bob, we should start our own company. So we did, and actually I've kept doing that, as Sangeeta pointed out. And again, when I did this in the 1980s, it really did not go over real well at MIT. A lot of people felt you should be pure academic and not start companies. But I just felt it was, if we were going to get things out to the world, we'd do that. So we've kept doing it. And of course, that's led to many different products. These are just a few examples, and I'll get to a couple more in a second. But they're used to treat almost every disease, including cancer, heart disease, and they're not limited to medicine. One of the key challenges, and when even in the Nature paper, we made particles that were of nano size. But one of the key challenges when you do this is sometimes the molecules stick together and macrophages can eat them. So working with Vladimir Torchlin and others, we took polyethylene glycol, which is very hydrophilic, and we coated them. And then you can even put targeting molecules on. Omid Farrokhzad, when he was working with me at the Koch Institute, started doing that. And I just thought I'd show everybody a video that Nova, the TV show, made on our work, where they were doing some filming a number of years ago on making things smaller. I don't know if the sound is on.
in a plastic
let me just
drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Now, a lot of the clinicians I work with at the Koch Institute tell me that it doesn't really blow the cell up that way. But you get the idea. And of course, I think, and of course I'm prejudiced, but I'm not going to use my own quotes, that delivery is a huge problem. And it was a huge problem for small molecules. It's an even bigger problem when you use large molecules. And here is Inder Verma, who, when he was president of the Gene Therapy Society, saying there are only 3 problems in gene therapy: delivery, delivery, and delivery. And our colleague, Phil Sharp, who was once head of the Cancer Center and member of the Koch Institute, pointing out that delivery, again, is this giant problem. So these were the sort of the tenets of the technology. And but what's happened is that really the Koch Institute and now the Marble Center ended up so many students making tremendous contributions. So Phil and others started Alnylam, a local company. Dan Anderson, who is a member of the Koch Institute, started out as a postdoc with me and did unbelievable work in the lab, came up with the first high-throughput synthesis approaches to make polymers and lipids. We've looked at all different types of materials. And we had a student, Kenna Kink, and when Alnylam started, he joined it. And I was on the scientific advisory board from the beginning. Dan was a consultant for many years, and Kink led this work, and that led to Onpattro. That's the very first siRNA drug that was ever approved. It was approved actually in 2018 to treat ATTR amyloidosis, and that. But again, that really would not have happened without the Koch Institute and the Marble Center. People probably know, as Sangeeta mentioned, that I was involved, along with others, in starting Moderna. Noubar Afeyan, actually, who was an MIT graduate, was another co-founder. And of course, this, I think, by now gets pretty historic. When COVID hit, actually, Moderna, who hired a number of students from our lab and the Koch Institute, really in just less than 2 months took this thing from a concept to the first trial, the first in human trials, and ultimately this is a product that was created, the COVID vaccine, which has been used by hundreds of millions, probably, I mean, I guess by now many billions of people around the world. And it's really that and the BioNTech Pfizer vaccine, have been by far the most, which is also a nanoparticle, again, based on some of the work that I've already mentioned, all over the world. And again, that would not have happened without some of the work I mentioned and some of the work that came under the NCI grant and later the Marble Center and the people that did it. And in terms of cancer, this is just a recent work. Much of it's been published by Moderna and Merck. And if you look at this is a potential way that cancer may be treated by really using the same principles and this is a phase 2 trial, 157 patients with very advanced melanoma. And you can now, they're creating a vaccine. In a sense, this is more of a therapeutic vaccine where the tumor mutates, and you can select up to 34 mutations known as neoepitopes, and you put it in the nanoparticles, exactly the way it was done in the COVID vaccine. Keytruda is probably the best, biggest selling drug right now in the world, and what they did is it's an immunotherapy drug. Half the patients get Keytruda, half get that plus this. The risk of recurrence or death was reduced by 44%, 2 years, 49%, 3 years, 49% at 5 years. And now Moderna is in about 8 different clinical trials, very advanced clinical trials, phase 2 and phase 3. with different kinds of cancer treatments with mRNA. BioNTech is doing it, and other places are doing it too. So there's a potentially big hope for the future. One of the challenges in this, and really any vaccine, particularly if you go to the developing world, is that people don't come back for a second shot. Even in this, you're actually giving 9 injections over every 3 weeks. And so Bill Gates came to see me at one point, and because of the developing world issue, asked, could we ever make what I'll call a self-boosting vaccine? And that would mean you get pulses at different times like you see here. So Anna Jakulenik, who this picture Sangeeta showed before, came up with a way to make a new printing approach. This actually even involves pico technology, because you have to do a pico filling in these little particles, and you seal them up. But they're like tiny eggshells, so you can make them break at precise times by changing the chemical composition. And so we came up with a whole printing approach, and by changing the polymer composition with the FDA-approved polymer, polylactic glycolic acid, by changing the ratio of the lactic to glycolic acid, by making it an acid versus an ester, by changing the molecular weight or adding a buffer, you can make these break at almost any time. And so this has been something that we're hoping will be used in the developing world and here for many things. To that end, BARDA came to see Anna and asked, could you ever make a portable printing machine that you could make little microneedle patches? And I just want to show you what a microneedle patch looks like. So our vision with the Gates Foundation is that someday you'll have these little spokes. These are little needles. And when you put them, it's like a Band-Aid. You can see this here. And so the idea that we had is that someday what you could do is put the little nanoparticles in little needles. And then you'd have this little patch. But the needles go in far enough so that you'll actually get the nanoparticles, but not so far that it causes hardly any pain. So it's a pretty painless approach. And so the hope is that we could ship these all over the world. And to that end, Anna and our team actually made a tiny little printer that you could just print these little patches in. It's published in Nature Biotechnology. And it worked actually very, very well. You actually, if you look at the receptor binding domain, you can compare the standard, let's say Moderna or BioNTech system, looking at pre-boost and boost, to what happens with the microneedle patches. And they're pretty comparable. And we saw no change in stability over 6 months, even at room temperature. Again, Anna did a lot of work on trying to figure out how to make these very stable by playing around with different lipids. I think the future, actually, I did make a slide on future, but I think Sangeeta did so much better job than I could ever do. And of course, the people that we have here at the, at the Marble Center, there's been great work done by Sangeeta and Angie Belcher on creating all kinds of new sensors. And Sangeeta also mentioned theragnostics, the idea that you could both sense and deliver things. And of course, artificial intelligence has a tremendous effect on all that we do. And I think, again, Sangeeta's example of what Dan and I have been involved with has been very, very, very illustrative. And of course, it's not just Dan and I that work on drug delivery. We're thrilled to have Paula Hammond's done incredible work on developing new layer-by-layer systems, and Michael Birnbaum's been doing some wonderful work on immune approaches, and we had Daryl Irvine before doing great work in that area. So I just wanted to share with you some of those things, and really it's been an honor, and I can't thank you enough, Kurt, for helping, and actually the leadership of Sangeeta, and Tarek has been incredible, and Matt, I think, has just done an amazing job as head of the Koch. So thank you all very, very much.
All right, I think this is now time for our panel. If our panelists can come on the chairs here, and thank you.
There we go. That was wonderful, inspiring, amazing. For those of us who don't live in the nano world, every time I get introduced to it, I just, I just have this sense of thrill that the world that we can't see is so rich with detail, but also rich with possibility. And I have to say, it's been one of the great privileges of my being at MIT is to be able to see people unveil that. So anyway, welcome everyone. And thank you for joining us. Congratulations to the Marble Center. Kurt, I mean, I've known you a long time and have never managed to thank you enough for what you've enabled here. So thank you very, very much. So welcome everyone, and thank you for joining us. I'm Susan Hockfield. I'm the Whitehead Professor of Neuroscience at MIT. And many of you know this, but I served as MIT's 16th president from 2004 to 2012. And I guess not news. I was the first woman and the first life scientist to serve in the role. People ask me about that. I think being the first life scientist was even more disruptive than being the first woman, but there was a lot of good work to be done. One of the really brightest highlights of my time as president was a chance to encourage and accelerate the convergence of life sciences, engineering, and the physical sciences at MIT's Koch Institute for Integrative Cancer Research, and really now across all of the institute. So today's discussion is about something many of us care deeply about, which is how breakthrough science in a place like the Marble Center drives new therapeutic and new diagnostic approaches that can reach patients. These aren't just interesting esoteric scientific experiments, technological experiments. These are about changing lives of people. Today's discussion is really, we're going to try to explore not only the scientific ideas behind new therapies and new companies, but also the less visible parts of the translational journey. You'll hear a common theme throughout the conversation. Great science is absolutely essential, but it's not sufficient. Translation requires us to connect, collaborate, adapt, and often a willingness to rethink what success actually looks like at each stage. So let me introduce you to four fantastic panelists who join me up here today. And I'm going to make, I apologize, but only extremely brief introductions. Each of them has a career and a set of discoveries that would exceed the whole hour if I were to give any of them in appropriate detail. So I apologize that time constraints prohibit a full set of accolades. So first, Vadim Dudkin is founding CTO at Souffle Therapeutics. Give a wave so people they can't read. We don't have name tags, so you have to wave. And he has more than 20 years of experience in the field, beginning with his career at Merck, leading a team that was focused on siRNA conjugate research, to serving as global head of RNA and targeted therapeutics at J&J with worldwide platform responsibility for RNA medicines, protein conjugates, ADCs, and targeted nanotherapies discovery portfolio. Noor Khani is CEO and co-founder at Matrixome Bio. As founding CEO, she's the inventor of the company's core technology. She raised the company's seed round and defined its strategic vision. Noor received her PhD in immunology and did her postdoctoral training in Richard Hein's lab at MIT, in the Koch Institute for Integrative Cancer Research, where she was also named a 2025 Termier Fellow. Noor has over 15 years of experience in oncology and extracellular matrix biology. Pete DeMuth is the founding scientist and chief scientific officer at Alicio Therapeutics, where he leads the development of technology platforms for cancer immunotherapy from early research all the way to the clinic. He received his PhD in biological engineering at MIT working with the labs of Daryl Irvine and Paula Hammond at MIT's Koch Institute for Integrative Cancer Research, which is just across the street from us here. Pete has spent over 15 years working at the intersection of oncology, immunology, and material science. Victor Edelsteinson is the director of the Gerstner Center for Cancer Diagnostics at the Broad Institute of MIT and Harvard, which is where we are, due to some complicated physical issues in our building across the street. But he leads, here at the Broad, he leads the blood biopsy team, which is a multi-institutional collaboration to profile cancer genomes directly from blood samples, which is one of the hottest areas in diagnostics today. Victor received his PhD in chemical engineering at MIT from Chris Love's lab at the Koch Institute. He's a faculty co-founder of Amplifier Bio. We just heard a little mention of it, a brief mention of that company, along with Sangeeta, Bhatia, and Chris Love, who are also members here. Amplifier is a startup developing a transformative technology in the field of liquid biopsy. So I would ask you, please join me in welcoming our panelists. It is quite a star-filled cast. So to start, I want to ground us all in the core problem each of you is trying to solve. These are, people who are, doing great, just kind of exploratory research, but definitely doing it with a purpose. So let's start with you, Pete. What problem are you trying to solve at Elicio, and why is your approach uniquely positioned to address it?
Yeah, thanks, Susan. First, I want to say it's amazing to be back here. This community and this center and this group of people have shaped me professionally and as a scientist in so many ways, so it's wonderful to be back. Great honor. So I'm currently the Chief Scientific Officer at Elicio. The Elicio story begins about 15 years ago, some work done in Daryl Irvine's group. We're really trying to figure out why cancer vaccines, specifically peptide-based cancer vaccines, were not living up to the promise that we had all envisioned for them. And one of the first questions that we started to understand was where these molecules were going after they were administered. Sangeeta gave a great introduction that size and shape and all these features of nanomaterials really determine where they go in the body. And so one of the things we early on discovered was that these molecular adjuvants and peptide antigens, after they're administered, they weren't getting to the immune system where it's orchestrated in the body, the lymph nodes. We've talked about that already. Several speakers have mentioned it. So the Elicio technology was originated in Daryl's group, and it's essentially a way to reprogram the way that these conventional components of peptide vaccines move in the body after they're administered. And it uses this very interesting mechanism called albumin hitchhiking, whereby the components are modified to bind to albumin, this common protein in the tissues, which then serves as a shuttle to carry them into the lymph nodes, deliver them to immune cells. And it turns out that this is really important for generating robust, high magnitude, high quality immune responses. And Elicio is now advancing that for applications in oncology. We've run a few phase 1 trials. We're in the midst now of a phase 2 trial with a readout in the first half, which is very exciting. So that's the problem that we're trying to solve, and the platform technology that originated here across the street that we're using to do that.
Yeah, the theme is hacking the human, as one of my colleagues has called it. I think that's Angie's phrase. So Noor, tell us about Matrixome.
I'm going to start, Susan, by thanking the panel, the center for having me here. I see a lot of mentors, advisors. 5 years ago, I was sitting in the audience across as an associate member at the Broad, and I was a postdoc and a research scientist at MIT for 8 years at the Koch Institute. So I co-founded Matrixome 3 years ago with Richard Hines, my academic advisor, and with Sangeeta, Bhatia. The vision that we started the company on was that we were interested in leveraging the extracellular matrix as a therapeutic target across chronic diseases. So the matrix is a meshwork of proteins around all cells of the body. It's present in healthy tissues, but it's also present in diseased tissues. And in diseased tissues like tumors and metastases, this matrix is fundamentally altered. There are proteins that are specifically and abundantly present at these sites. And at Matrixome, we're leveraging these specific proteins as therapeutic targets. The unmet need we're trying to solve is advanced stage metastatic solid tumors. As you all know, that remains the largest unmet need in oncology. Despite all the successes in the field, even today, 90% of cancer-related deaths happen when cancers spread. So clearly, cancer cell-targeted approaches that have been working for many years don't work in the advanced metastatic context. We believe that the matrix offers a really powerful alternative to deliver therapies and to modulate cancer. Why do we believe that? In advanced metastasis, typically therapies don't work because cancer cells evolve and they mutate, and cancer cell-associated targets like cell surface receptors typically downregulate, and that's because cancer cells themselves are heterogeneous. The matrix is conserved in disease tissues. There are specific proteins present across solid tumors and also in metastases, and we believe its ability to not downregulate and not be subject to mutation makes it a very stable target. So Matrisome's lead assets are matrix-tethered radiopharmaceuticals. We are using small protein-based binders called nanobodies against disease-specific matrix proteins to deliver a variety of disease-modifying payloads. When we were at MIT, we collaborated extensively with professors across the ecosystem to deliver immunomodulators, which could turn cold tumors to hot and also have really strong impact on efficacy. But currently, the company is focused on making first-in-class matrix-tethered radioligand therapies. The idea is to deliver radiation specifically to tumors and metastases. So we're combining this modality, which is radiation that has worked in oncology for decades, with a new class of targets with a very differentiated biology. And this drug, as I mentioned, has the potential to be pan-tumor and pan-stage because matrix is conserved, the targets we're going after are conserved across tumors, across patients, and present also in metastatic lesions.
Wow, that's incredibly exciting. Being an extracellular matrix maven myself, we'll talk later. Victor, tell us about Amplifier.
Well, it's an honor to be here. MIT community is incredible. When I started my lab, I wanted to be as close as physically possible to MIT here at the Broad. So liquid biopsies could enable cancer detection and monitoring, but there's often very little tumor DNA in the blood. So little that it's difficult to detect the earliest stages of cancer and residual cancer that may be left behind after therapy. Now, most of the field has focused on developing better tests, but we believe there are physical limits on what tests can detect. If there's not tumor DNA in a blood tube, no amount of sequencing or machine learning will be able to overcome that. So together with Sangeeta, Bhatia, Chris Love, and the talented members of our labs, we developed the first priming agents for liquid biopsies, which are injections given before a blood draw to transiently slow the clearance of cell-free DNA from the bloodstream and allow 10 to 100-fold more tumor DNA to be recovered. And the idea of giving an injection to make a diagnostic better is not new. It's been done hundreds of millions of times for imaging scans, but our approach is the first of its kind for liquid biopsy, and we've started a company, Amplifier Bio, to advance that to patients.
Fantastic. Vadim, tell us about Soufflé and give us a hint where the name came from. The other names I kind of can figure out, but Soufflé, I'm mystified.
Maybe Bob is the better person to address that. But maybe I'll start by saying that we are incredibly lucky at Soufflé to have not one, but two Marble Center faculty among our founders, so Bob Langer and Dan Anderson. Our goal is to enable targeted delivery with receptor-mediated uptake to any type of cell in the body. And so that kind of, I think, the vision with which the company was started. Those of you who have been in delivery field, I think, would agree with me that ability to identify receptors and pathways that drive effective, safe, and potent uptake into cell types specifically probably has been one of the key drivers that enabled translation of advances in the field into benefits for the patients. And maybe in the siRNA specifically, discovery of ASGPR and GalNAc-mediated delivery probably created multiple drugs that are already on the market and probably over 100 billion in value over just the next decade or so. So with that, there's been a lot of effort to take existing receptors and figure out if we can use them to delivery of different cell types. And I think if you look at the whole field, there's still ASGPR is the only receptor that really drives selective and potent delivery to a single cell type that has been validated. So 4 years ago, we have decided to solve this problem with a systematic approach. And so what we have done is we identified ways to do screens on cells and even whole tissues to identify what receptors are on the surface and then how we can identify subsets of them that can drive productive delivery. So that became the foundation of our platform. We have translated that into ligands that have gone into development, into manufacturing. We have several programs, and so now we are at a place where we're expecting 2 products to get into the clinic in the next several months, and we have other programs in the wake.
Thank you, all of you. So a follow-up question. So I know you all received your degrees from academic labs, and we have this just curiosity-based desire to understand things. So I want to know for each of you, what was the moment when you realized that you had found something that could actually help patients, not just produce another publication? Noor, why don't you start?
So I actually remember the day when we saw our first images. We were doing experiments at MIT in the imaging facility at the Swanson Biotech Center to see if these matrix-specific nanobodies that we were making can actually home specifically to disease sites, to tumors and mets. So I think for us, for Richard and myself, this wasn't a moment, it was a journey. We were very excited about the data we saw. We started to present it at floor meetings, at the KI retreat, and we got really positive feedback from the faculty in the ecosystem. And I remember this one KI retreat where 3 different faculty came and spoke to me and Richard and said, "This is great, you should start a company." And I think that was the moment where we felt like this is something we can take forward. And the idea that we can target the matrix to help patients is, I think, something that Richard had pioneered and had worked on for decades. So this company is really, in some ways, taking that mission forward.
Great. Go ahead.
Yeah, this is a great question. And I think it's a great question because we're all scientists, and so the data is so exciting. It never stops being exciting. Like this example, I also have 2 moments where I can viscerally remember learning about results that made me convinced in the promise of what we were doing. The first was some of the preclinical studies where we were developing our therapeutic candidate for mutated KRAS. And we had designed a series of candidates. We were evaluating them in mice. And one of our scientists had texted me an image of an ELISpot plate, which was blowing up with color. And for the immunologists in the room, you know that that means that there's a profound immune response. So at that moment, it was just a very memorable realization that this could be applied in the KRAS space. And then the second one that I'll mention very briefly was as we were analyzing some of the data from our first phase 1, where we were trying to understand the correlation between the strength of the T cell response that was being induced by our therapy. And this was later in time where we had some follow-up, longer follow-up on the patients, and we were able to look at the clinical endpoints that are really meaningful for this patient population, disease-free survival and overall survival. And we started to see this very clear correlation between the characteristics of the immune response that were generated and the clinical benefit that was apparent in the patients that we were studying. And that was another moment of crystallizing realization that this could become something that really helps a lot of people.
That must have felt incredible.
Yeah, these are moments that you probably remember. You know, I'm sure people in the audience have moments like this that they'll remember. So.
Yeah, so I think, of course, there's nothing like getting your first in vivo result, right? And so that's one part of the story. But the other part of the story for me is if you have an aspiration to build platform company, you have to be able to demonstrate that your platform actually works and it's not a one-off. So we have, we built the platform, we have run our screens on our first cell type, we built the ligands, we conjugate them to a nucleic acid, and we saw that we can selectively silence a gene for the first time using that new delivery system. So of course that's incredible. But then the question for all of us was, can we do it again? Right? Is it a real platform that can be integrated and can be reproducible? And so then we did it again, and then we did it again. And I think at that point, we were starting to feel like it's not only a one-off discovery, but we have a platform that can continue to produce these results, and ultimately, if we're successful with our clinical trajectory, we can help a lot of patients.
Fantastic.
Well, so my team has done a lot of DNA sequencing. We've developed tests, including Maestro, that Exact Sciences, now Abbott, will commercialize this year. It'll be one of the most sensitive liquid biopsy tests for minimal residual disease. And so we've seen the power of DNA sequencing, but also the physical limits of what can and can't be detected. And for us, the aha moment was really when we applied these really sensitive sequencing methods to blood samples from mice that were collected immediately before and after administering these priming agents. And what we saw was that we could convert samples that were otherwise undetectable to detectable, and in mice at tumor burdens that were lower than we could otherwise detect. And we also saw much more of the tumor genome represented in the blood draw. And Sangeeta showed that nice graphic, the genome representation in the blood draw, and that's when, remember, we had a phone call, this was during COVID, just how excited we were about these results, and just seeing the impact that this could have for many types of liquid biopsy tests.
Yeah, terrific stories, I have to say, it gives me chills. So a follow-up question, and you don't have to answer this, but does any of you want to talk about whether you've ever had to pivot the original scientific idea or the original purpose to make it more of a product for the company? So in other words, did everything you had initially conceived make it as part of the translation story? And I guess the background of my question is, as a scientist, when you're just doing the science, you just kind of follow the curiosity, right? But when you're actually aiming for a real product, a therapy or a diagnostic, you have a very different orientation. So if any of you don't all have to say this, but if there was a moment when you actually had to pivot to, adjust to the goal that you had for the company, not just for getting your NIH grant.
I can start. Maybe not exactly a pivot, but there was a moment, I think, in our trajectory where when we figured out that the delivery side really starting to work and is fruitful, we could enable a whole spectrum of delivery systems with this approach, right? And so initially when we were thinking about what the future trajectory of the companies, we saw a whole spectrum of systems that we could power with our ligand receptor pairs. But at some point, of course, we realized that now is the time to start thinking about the products and the first products that we're going to take to the patients. And so maybe it's not a pivot, but I think there's a point where you really need to focus. And so that focus for us became siRNA, and that's going to be our first several products. We still have the broad picture in mind, and we're working and collaborating on that. But I think there's a moment where you need to figure out what your first product is going to be.
I'm happy to go next. I think similar to Vadim, we didn't have to pivot, but Matrosome's core technology was a platform technology, which could be used to deliver a variety of disease-modifying payloads. And when we started the company, we were excited about multiple options. But as we listened to feedback from investors, from the market, we had to focus and make choices. And that allowed us to then be very focused on taking one part of the platform forward. And I think also to Vadim's point, you have to show that the platform works by showing that one asset works, which is the approach that we're taking at Matrosome at the moment. So doing everything is very exciting, but sometimes not capital efficient. And so that's the choice that we eventually made.
It's got to be a hard decision. Pete, why don't you?
This is turning into a really thematic observation.
Yeah.
I think the excitement of a platform is great. But there's also a lot of pain in terms of making that decision of where do you go, how do you use it. And I think there's always this desire to go everywhere, but that's not feasible. You do have to make hard choices. And I think that the thing that keeps us up at night early in those early days is, have we made the right decision? Have we chose the right place? But that's also the beauty of a platform technology, is that there are so many opportunities. You have a lot of things that you can move and pivot to. And while we don't necessarily have a pivot story, we do have this shared experience of needing to focus, needing to prove something works once, and then move on to the other areas that we'd like to explore with the platform.
Yeah. Go ahead.
Well, I'd say one of the benefits of working with folks like Sangeeta and Chris is just they're always thinking not just what will work in the lab, but what could really make it out there into the world to benefit patients. And just really grateful for that, because I think it's kept the team really focused on approaches that have strong clinical precedent, manufacturability, that could conceivably be used in humans someday. And so even though our work together has taken various twists and turns in the kinds of approaches we've tried, the wild thing is what's ultimately now being advanced to patients is one of the kind of early and simple ideas, right? That just works in mice, and we're taking to see if it works in humans.
Fingers crossed for all of you in that domain. So, we all understand that translation, as you just said, requires a lot more than great data. You need capital. You always need a little bit of capital to run the experiments, but it's very different when you're talking about clinical trials, right? Talent, institutional support, strategic collaborations. I mean, there are so many things, so many gaps that are different from what happens when you're just pursuing science. That you've gotten to get from invention to impact is a big, challenging journey. So I want to expand a little bit more on collaboration, since the idea of collaboration is really the central thesis of the Marble Center. It's one thing to gather a bunch of people who are just, superb scientists or even superb at translation, but I think during the translation piece, it really takes a lot more of collaboration, considering where we all come from, which is with PhDs out of graduate school, right? So if you can, why don't we start with Victor? So what's the single most valuable contribution an academic partner or research center like the Marble Center can provide to your startup idea?
Well, I've been fortunate to have a long-standing collaboration with Sangeeta, Chris Love, the Marble Center for the past 7 years now, certainly with Chris preceding to my graduate years. And I'd say the single most valuable contribution is really the people. I mean, what an incredibly talented, smart, creative, and collaborative set of individuals here. It's really been their wisdom, expertise, and drive that's allowed us to tackle very important problems and difficult ones in cancer diagnostics. And the wild thing with our work has been the parallels to with a lot of focus in nanomedicine has been how to get things into tumors. Our question is sort of flipped, right? How do you get things out? And how do you get tumor DNA out? And how do you detect that better from the blood? And so it's just been incredible, all the brainstorming and collaboration with the people of Sangeeta's lab and the Marble Center, just couldn't have done it without them.
So actually, that's kind of this is amazing. It's leading to exactly the questions that I have written down, which is, so one question that I think you may have answered already, but we can come back to you. But what's the single most valuable contribution that a partner has made to your progress? I mean, you were just, you don't have to answer it, but if there's some particular moment of collaboration that really broke through the wall for you, it'd be interesting to hear about it. Anyone?
I mean, I think I can echo some of what was just said in that we had the great fortune of working with Daryl as he developed the technology. And then there was a point at which it was transferred into Alisio for the purpose of developing it for products. And then manufacturability became a big focus, and how do we push this forward into the clinical arena. What was great about that partnership, one of the great things about it, was that the big sky thinking was still going on with our academic partner, with Daryl, with his group. And so the platform was able to be pushed in a variety of different directions to unlock potentially new applications, while we did sort of the work behind the curtain to kind of get the platform ready for scaled manufacturing, for transition and translation to the clinic. So it's a very symbiotic and very complementary set of roles that we each performed to make the most of what was a really exciting and promising strategy.
Great example. Anyone else want to talk about that transition?
I just want to give my perspective maybe. It's hard to talk about a single contribution because collaborations are so multifaceted. And I think in my experience, I had, when I was at J&J, I had probably tens of collaborations in my teams, and they have contributed tremendously. I was lucky to have some of my teams be part of the Koch Institute Janssen collaboration over the years, and it's just so rich, right? There's so many components of these programs that come for these interactions. It can be technology, it can be targets, it can be means of assessing delivery, imaging. There's so much. So I'm struggling to think about a single thing because it's just hard to focus on one thing.
That's actually a great description of how things actually get done. The next topic is scale up. Because when you're just doing research, scale up is not really an issue. But if you're doing something that going to have some impact broader, you have to scale it up. So this is often a place where translational enthusiasm meets industrial reality. And so you can have a beautiful little thing, but you've got to be able to scale it. So something that works in a small academic setting may represent very different problems when you need consistency, reproducibility, cost discipline. I mean, this is just a whole set of challenges that, as a lab scientist, that's not going to be, none of those are going to be on your mind. So I'm wondering if, Bhatia, you have a story that you can share about how assumptions from an academic lab and an early stage idea turned out not to hold up at manufacturing scale.
Yeah, I've been part of bringing many things into a clinic, and of course you have to think about it not just in terms of costs, but also in terms of ultimately the risks that you put into the product. And so maybe one example is with our current programs, it's conjugation. So, as a reference point, bioconjugation has been one of the probably cornerstones of my career. I've been working on it for over 20 years. I've been lucky that I had the privilege of leading teams that solved many problems using novel bioconjugation approaches. So when we start building Soufflé platform, working together with Dan and Bob and other founders, we built a whole suite of methods, right? That they're novel, they're highly efficient chemical chemoenzymatic methods that allow us to quickly put these things together, take them from early idea into in vivo testing really fast, and so that was super exciting. State of the art. Now, we started developing our first product, and I have to think long and hard about, do I want to use the most exciting, novel approach, right, that is state of the art, it works great in the discovery setting, but it has impacts, right? There are regulatory risks because these things are not in clinical products yet. There are manufacturing risks because you have novel entities, linkers, things that need to be manufactured, some of them are explosive, so you need to balance all of that. And the brutal reality of drug discovery and development is that the risks tend to compound. So you bring several risks on the COGS side, and you bring risks on regulatory or safety. And at the end of the day, you're running into a spot where these risks will compound, and so that is less likely that your molecule will reach the licensing. So the solution to this is you focus where the risks are that are important for you to assess. Usually it's a biology risk or it's a platform. And then you take all the other risks out. So we ended up going from the fanciest, most sophisticated conjugation technologies to using things that are in already approved drugs and have been in those for decades. So that's my example.
Interesting. Thank you. Thank you for sharing that. So does anyone else have a, what I call scale up isn't easy story? Or for the rest of you, just, you could do tenfold, 100-fold, 1,000-fold.
So the example that I think comes to mind is a little bit different, but similar themes in that Alyssio's candidates are built upon the types of molecules that the industry has already developed methods for making at large scale, peptides, nucleic acids. But there was a lot of thinking that needed to be done about the modifications that were novel to our platform that enabled the lymph node targeting mechanism to be successful. And so fortunately, Daryl and his group did a great job of developing that. But as we brought it into the company, we started to look at each of the reagents that we're using with a very strong magnification, so to speak, wanting to see how any of these things would trip us up as we got to scaled manufacturing. And the example that comes to mind is the polyethylene glycol. In the academic sense, we're using readily available, very cheap, very affordable, heterogeneous polyethylene glycols, which have a number of different molecular weights. Perfectly suitable for the academic world. But as we started to think about scaling this and putting it into a clinical trial, potentially with multiple agents that would be very similar, we started to imagine this convoluted mass problem where many agents would have very complex mass profiles. And so the decision we made was to use discrete polyethylene glycol, which is more expensive, but also readily available, manufacturable. And what that did was allow us to move from an end product that would have multiple different discrete masses to a single molecular entity that would have one mass. And so that eliminates a lot of complexity on the regulatory side, on the CMC side, and ultimately gives us a lot more control over the product as it goes forward.
Fascinating. Here's another kind of practical question or a plea for advice from all of you. And it's something that, we in the academic world, again, don't really think about very often, but how early should a founder or the founders begin thinking about manufacturability? One thing in a test tube. Or
Early.
You're early.
Early, right away.
No, is it right away? I mean, go ahead.
I mean, thinking, absolutely thinking.
Yeah.
Having a finalized plan, not necessarily right away, but I think the earlier you start thinking about that, the better off you will be in making good decisions and then eventually settling on the things that will make you successful as you scale and manufacture and move forward to translation.
Yeah, so we're a preclinical stage company, so we're not, solving manufacturing problems, but I absolutely agree. We are already thinking about developability analysis, de-risking manufacturing, and making choices that are just, de-risked from the context of things that have been done before, things that are, cheaper, faster, or you can find CDMOs to partner with. Our drug is a biologic, but it also requires conjugation. And similar to some of the examples shared, initially we were using a very unique enzymatic conjugation method that works really well, is very quick, can be done in the lab, but it's very hard to scale when you think about advanced manufacturing. And so we moved away from that to go to very simple methods that have been done before. So, we're maybe 2 years away from the clinic, but already thinking about a very clear plan to de-risk both developability and manufacturing for our drugs.
Maybe I'll add. So I spent some years helping make investment decisions when I was at Johnson & Johnson with a venture team. And a lot of times the decision comes to not really manufacturability, but having a path, right? And so if you're trying to start something new and it requires manufacturing, at the very least, you should be able to produce in vivo material, right? And then you should have a path to NHP material, and that's kind of how we view these decisions. And I can't tell you how many times just making enough material for a well-controlled mouse study was hard, right? So you don't need to have a GMP path and full analytical control strategy, but you need to have an idea of how you're going to make the molecule and how you're going to make it for a monkey study. I think that goes a long way.
And yeah, I would add, really in the academic lab, that's not to say it should stop the pursuit of science. There's all sorts of experiments we try that approaches that would never feasibly make it into the clinic, but still with the goal of developing things that will benefit patients. The more we can focus early on approaches that we think will have clinical tractability someday, the better.
Okay, we're going to wrap things up. Yep. Saw me looking at my watch and realized we're on the same. We are on the same wavelength. Okay, so this is again just out of immense curiosity, but I just want to ask you all that as you look ahead 10 years, if you can. I mean, science is a little unpredictable. What do you think will be the most transformative impact of nanomedicine? I mean, we are in a different world all of a sudden. And as you imagine where it's going to take us, where do you think the biggest impacts are going to be? Or one?
I think it's precision. Being able to eliminate side effects, being able to deliver medicines more precisely to get more effect by doing those things, to open the therapeutic window in ways that we haven't been able to before. We've talked about diagnostics, making treatment decisions, all the things that will allow the practice of medicine to be more precise. I think it's going to happen at the intersection of fields, which is why centers like this are so great. But I hope the impact in 10 years, we'll be sitting here saying many diseases that were very serious, hard to treat, high unmet need, are now either manageable or curable through the advances that this type of technology have brought.
Noor?
So I agree. I was thinking about, just new medicines, new devices, and scalability across indications, across patients for nanomedicine. I think precision is definitely something that's, what can work with nanomedicine. I think one thing that nanomedicine can really impact is repurposing drugs that haven't worked systemically for dose-limiting toxicities. And I think nanomedicine can really make significant breakthroughs in the next 10 years in that field.
Yeah, so for me, I guess my dream is that this field is, I think, at an incredible place right now where there's a lot of advances on the engineering side, different delivery systems, there are different ways of let's say you're in genetic medicine space, packaging payloads. There are different types of payloads. The new ones are coming and getting described almost every day. At some point, I think there will be a convergence of this field based on the idea that what we're trying to do is we're trying to deliver instructions into the cell. And so it becomes an engineering and information challenge. I think when we figured out delivery, we figured out how to drive the molecular biology of the cell in a way that will alleviate the disease. And then the problem becomes, can we understand the genetic background? And then we use the technology to fix it, basically by transferring instructions into the cell. So that's kind of, I see the convergence point of many disparate efforts that are going on right now.
Pete, you get the last word, and I'll close it out.
Well, I'm excited about early detection and interception. I think having the capability to detect cancer early, whether that's through liquid biopsy with advances in imaging, the various approaches we've heard, to determine its molecular profile in a minimally invasive fashion that could guide precision intervention through targeted drug delivery, through personalized tumor vaccines, a number of approaches which the earlier we can administer those, the more effective they could be.
Fantastic. I want to thank you all. This has been an amazing glimpse into the future. And I want to thank all of our panelists for their honest and insightful discussion. But mostly, I have to tell you, it still thrills me to see the frontier of scientific innovation. And as I said, I'm the convergence queen. I just love it that you all are, pulling in all the different pieces that you can find to improve lives and save lives. And I'm incredibly grateful to you for being joining the center, but also for doing what you do. So thank you all very, very much.
Thank you, Susan.
So much for our wonderful panelists and for Susan for moderating this panel. So I want to call Dr. Matt Vander Heiden next to come up here and give us his closing remarks. Matt is a director of the Koch Institute at MIT, the Lester Wolf Professor of Molecular Biology, and a member of the Broad Institute. He's also a practicing oncologist instructor in medicine at Dana-Farber Cancer Institute, Harvard Medical School. Matt is also an incredible mentor. He is involved in so many different types of startups and ventures, numerous awards. He earned his doctoral medical degrees from the University of Chicago, where he worked in the laboratory of Craig Thompson. And he completed residency in internal medicine at Boston Brigham and Women's Hospital and Hematology Oncology Fellowship at Dana-Farber Cancer Institute, MGH. Matt?
Thanks so much, Tarek, and congratulations to you on a really wonderful event. So I've had the privilege of being director of the Koch Institute for 5 years. This is the 10-year anniversary, which means I have to thank Tyler Jacks and his visionary leadership for establishing the Marble Center with Kurt 10 years ago. But I have been at the Koch for 10 years, so I can also attest to the fact that the Marble Center really represents something truly special. As you've heard today, it's a place where bold ideas turn into transformative advances that help cancer patients, and really we heard non-cancer patients as well. The advances that we've heard about and that are coming in early detection, more precise treatments, better delivery methods, it's really exciting. What is unfolding, the momentum that exists, and I'm really excited to see what's going to happen as we look forward for the next 10 years. Now, of course, I also want to thank Kurt. I would be remiss not to thank him really express our gratitude for you supporting and founding the Marble Center. Your vision together with your wife, Kathy, was extraordinary, and it really made the advances and the exciting things that we've heard about today possible. I hope you feel the same sense of pride that I have in everything that has been accomplished over the last 10 years, and I hope you're equally excited about what the future holds. I think it's actually incredibly bright, and I'm going to the answer to Susan's question, I'm going to say I don't even think we can imagine what nanomedicine is going to do over the next 10 years. Sangeeta, I want to congratulate you as well and thank you for your incredible leadership for the center over the last 10 years. These things don't happen without great leadership. And then in closing, I'd just like to give thanks to a number of people, largely around tonight's event. I want to thank all of our speakers, so Sangeeta, you again, Bob, our panel, Susan for moderating. The discussion, the talks were all very compelling. I want to thank Tarek and the entire team for all their work to pull together tonight's event. I want to thank the alliance partners of the Koch Institute who are here tonight. That's Astellas and Sanofi, who have representatives. I also want to thank MIT Nano, and particularly the undergraduates who worked with MIT Nano to make Kurt a really special gift. And I want to thank the Broad Institute for hosting us. We're not across the street yet because we had a flood. It's almost ready to reopen the auditorium, but not quite yet. But fortunately, the lobby is open, the Koch Institute galleries. And so when the program here is concluded, I want to invite all of you to please come back across the street and join us for a reception where we can continue our conversation and also celebration for the past 10 years. Thank you all again for being here.